The small intestine absorption, particularly the outline for physio, will be section B, absorption, all the way through here. Now, putting that information into a diagram, the small intestine absorption, we have several sections. We have the length, which is bigger than 3 meters. The macrovilli, it's pointing to these microvilli. It increases surface area. The mucosa includes the epithelium, which is the enterocytes. That's another word for the simple columnar epithelium. It's larger than usual, just to show more detail. The goblet cells are located here. And also just keep in mind that the mucosa is here, and then also inside here, particularly the lamina propria. So for now, just to start off with the simple columnar epithelium, the potassium, chloride, and sodium, they travel through diffusion of channels. This section here is crossed by the apical membrane and basolateral membrane. So we could just put pretty quick the secondary transport here is the 2. Co-transport with sodium is the arrow, diffusion through channels, and the primary active transport. Again, the potassium, chloride, and the sodium will travel through diffusion of channels. The sodium particularly can join with the glucose for co-transport with sodium, or the amino acid with the sodium can go together via co-transport using the secondary active transport. Now the potassium, chloride, and sodium can continue on going to or getting absorbed through the primary active transport. The polar nutrients can then be diffused into the intestinal capillaries, which is here in orange. It's the other part of the mucosa, which is the lamina propria or connective tissue. The polar nutrients will then diffuse and enter the hepatic portal vein and then the liver. Or lipids will be turned into chylomicrons and enter the lacteal. The lacteal is just another name for lymphatic vessels. Keep in mind the goblet cells secrete mucus and H2O. Well, of course, H2Os follow solute. And that's it for this one. The villi. All right, small intestine absorption.